In this module, we'll look at the receptors that mediate the effects of sympathetic and parasympathetic innervation. Sympathetic and parasympathetic effects are mediated by different receptors. Receptors are proteins that bind to extracellular chemical signals and convert them to intracellular responses. The autonomic nervous system has two main receptor types: cholinergic receptors, which bind to acetylcholine, and adrenergic receptors, which bind to adrenaline and noradrenaline. Cholinergic receptors are further classified into nicotinic and muscarinic receptors according to whether they can be stimulated by nicotine or by muscarine. You may be familiar with nicotine, a chemical found in tobacco products, and less familiar with muscarine, a chemical found in the Amanita muscaria mushroom. Nicotine and muscarine bind exclusively to either the nicotinic or muscarinic receptors, and that is how these receptors were originally differentiated. There are also two types of adrenergic receptors, namely alpha and beta adrenergic receptors. Let's look at these receptors in more detail. Nicotinic receptors are ligand-gated ion channels, meaning that when acetylcholine or other ligands bind to them, a central pore or channel opens to allow ions to flow across the cell membrane. This leads to membrane depolarization, allowing the cell to propagate signals to other cells. There are two subtypes of nicotinic receptors: neuronal nicotinic receptors, which are found at autonomic ganglia, the adrenal medulla, and throughout the central nervous system, and muscle-type nicotinic receptors, which are found at neuromuscular junctions of skeletal muscles. Muscarinic receptors are G-protein-coupled receptors, or GPCRs. This means that they couple to G proteins, short for guanine nucleotide binding proteins, which are responsible for kick-starting intracellular signaling cascades upon ligand binding. Activation of GPCRs generates second messengers, which mediate various cellular responses, such as muscle contraction and gland secretion. There are three main types of G proteins. Depending on which G protein a GPCR is coupled to, the intracellular response generated will be different. There are 5 muscarinic receptor subtypes, M1 to M5. They couple to different G proteins and have different distributions in the body. The M1 muscarinic receptor can be found in glands and the central nervous system. M2 receptors are distributed throughout the heart and smooth muscle. M3 receptors can be found in smooth muscle and glands. M4 receptors are distributed throughout nerve cells, and M5 receptors are found in the CNS as well as other areas of the body. Like muscarinic receptors, adrenergic receptors are also GPCRs. There are two main types of adrenergic receptors, alpha and beta, which are further divided into 9 subtypes. Alpha-1 adrenergic receptors are predominantly expressed in smooth muscles, such as blood vessels of visceral organs. Alpha-2 adrenergic receptors are predominantly expressed in presynaptic nerves. On the other hand, beta-1 adrenergic receptors are predominantly expressed in the heart, while beta-2 adrenergic receptors are predominantly expressed on the smooth muscle of bronchioles and blood vessels that supply skeletal muscles. Beta-3 adrenergic receptors are predominantly expressed in adipose tissues and the urinary bladder. Alpha adrenergic receptors are generally associated with excitatory responses and smooth muscle and sphincter contraction, whereas beta adrenergic receptors are inhibitory and induce smooth muscle relaxation. The exception is that beta adrenergic receptors at the heart increase heart rate and cardiac output upon stimulation. The different distribution of receptor subtypes in the body allows for fine control of responses. In summary, cholinergic and adrenergic receptors mediate neurotransmission in the autonomic nervous system. Cholinergic receptors include nicotinic receptors, which are ligand-gated ion channels, and muscarinic receptors, which are GPCRs. Adrenergic receptors include alpha and beta adrenergic receptors, which are also GPCRs. These receptors have different distributions in the body and mediate different effects, making them possible drug targets. You should now understand how the brain and spinal cord communicate with organs via the peripheral nervous system using neurotransmitters and receptors. The sympathetic and parasympathetic nervous systems exert opposite effects to maintain physiological homeostasis. These are governed by the cholinergic and adrenergic systems.